Alright, so we're back with what should be the third and final section of our Chapter 1 lecture, moving on to the overall basic study of how scientists look at the natural world. And so one thing we have to remember about science is that it is the only observation-based way we have of finding out information about the natural world. You've probably all seen the scientific method before in a textbook, the steps that are involved, maybe an example that probably involved either a flashlight or a car not starting. For some reason, all the books use those two examples. But we're going to go through it again anyway just to kind of review this because this is a science course and because this is something which is based very, very much on the scientific method, we need to know kind of what that's about. So the very first thing that happens with the scientific method is that someone observes some sort of phenomenon. And it doesn't have to be something, earth-shattering, groundbreaking scientific discovery, cure for cancer kind of thing. It could be something we do in our daily lives. One thing that people don't realize is that all of us do the scientific method each and every day, probably multiple times a day. This is how we go about solving problems. So after the initial observation that, hey, look, my car's not starting or my flashlight's not coming on, there is then a question that's going to be asked about perhaps why won't my blue car start or why won't my flashlight turn on, something like that, right? So we have our questions. Now we're going to start looking at hypotheses. And a hypothesis, which I'm sure you've all been exposed to this term before, is just an educated guess. It's a proposed explanation. It's not a random guess because we tend to usually have some idea in the back of our minds about what might be causing the phenomenon in question. It's an educated guess, right? A possible explanation to our question that we asked back in step number 1. And if we have now our educated guess, we can start looking at predictions. Well, if I change this, then I should see that outcome. That would be an example of a prediction that you might see using the scientific method. Hypotheses are kind of the front and center part of the scientific method, right? We talked about what they are. We didn't talk about the fact that really you don't test every single possible hypothesis because you don't have the time or the energy or the money to test every possible hypothesis. So you really have to do a good job at narrowing it down. You got to find out what you think are maybe the 1 or 2 most likely to give you information about your question kinds of hypotheses that are out there. The 2 that are going to give you the best bang for your buck. Maybe the 1 that's going to give you the best bang for your buck. So no, we don't ever test every single possible hypothesis. That would be impossible. This again is a scientific method, so this is going to mean that we're going to have to leave out things that cannot be tested. We're going to have to leave out things that cannot be shown to be false. So supernatural causes for things, value judgments, what's right or wrong or good or bad or better or worse, those kinds of things not subject to the scientific method because they're not testable and they're not falsifiable. Right? Another term or another idea we need to think about here is the term theory. Right? This theory with a capital T. This is a theory in biology. Right? This is a scientific theory. If it has a capital T means scientific theory. Scientific theories have been tested thousands, tens of thousands of times, if not more, and so far have not been shown to be false. So things like Darwin's theory of evolution, things like cell theory, things like theory of relativity, those kinds of theories are more like principles or laws. Right? So in biology, we don't have laws. We don't have the principle of such and such a thing. We have theories. A theory is as close to truth as you can get in biology. Why don't we call it a law? Why don't we call it a principle? Those things have to be true 100% of the time. And living things are too variable to be 100% anything. Okay, so theory is about as close to a law as you can get in biology. Now, when you talk about a theory like an everyday theory, like, oh, I've got a theory about who's going to win the NBA Finals or whatever, that's a different kind of theory. That's more of like a hypothesis. That's theory with a little t. These kinds of theories with a big T, that's an important theory. That's a scientific theory. That one has already been tested. It's not a guess. It's something which has been tested many, many times and so far has not been shown to be false. All right, the next thing we have to do is to come up with some sort of test or experiment. And this is probably the hardest part of the entire thing, is coming up with a good, bias-free experiment that's going to give us good information about the question that we're dealing with. Okay? We're going to talk about controlled observations here in a second as well. And eventually, at some point, you come up with a conclusion. Now, typically during this process, you're going to generate 200 other questions which you could test. In the process of answering this one question, or maybe answering this one question, maybe not even doing that, you're going to come up with a bunch of other questions you might want to ask as well. Okay, so we're going to talk about a couple more of these terms. Now, here's our scientific method. We've got our observations, our questions, our hypotheses based on prior knowledge. We've got an experiment collecting data. We're drawing conclusions, and eventually going back and starting it again. Or if we're professional scientists and our results are worthy of such a thing, we might actually try to publish it. And so we have this thing in science called the peer review process, where if you want to publish something in a scientific journal, not like a blog or something, but a scientific journal, it has to go through a peer review process. And what that means is when you write up a manuscript about some experiment you've done, you send it off to the journal you want to get it published in, and they're going to send it off to maybe 2 or 3 other experts in your field to have them read it over and see if what you did was legit. And if it's not, for any reason, they're probably going to reject it. They might say that, okay, well, it's not bad. I just want to see maybe a couple of revisions, a couple of additional experiments, do some more stuff. Or they might say it's horrible, I never want to see it again. It's really, really tough to get things through this process. So by the time you actually pick up a scientific journal, Science, Nature, those kinds of things, you can be pretty sure that the stuff in there is pretty good. It's already been through a quality control process by experts. Okay, so that's the publishing part of things. All right, back to the scientific method and the experiment part of things. There are different types of variables you want to look at in a scientific test. There are dependent variables. There are independent variables. Right? So we want to kind of work out those terms first. An independent variable is something that the researcher changes, alters, modifies, something like that. Right? The independent variable is what you as the researcher actually change. So if we're looking at an experiment trying to figure out the best level of fertilizer to maximize tomato plant growth, fertilizer level would be the independent variable because that's what you're going to change to see what kind of effect might occur. That's your independent variable. The dependent variable is the outcome of that change. Another way of thinking about that is the dependent variable is what you measure. The independent is what you change. The dependent is what you measure as a scientist. So in our tomato plant example, tomato plant growth would be the dependent variable. As you change levels of fertilizer, as you change the independent variable, you can measure changes in the dependent variable, the outcome. Okay? Now, for the best experiments are the simplest ones. Right? The best experiments are where research takes a single variable, one little variable, and changes that to see what occurs with the dependent variable in the end. In reality, is that very easy to do? Probably not. Even if you're in a lab or greenhouse, really controlled conditions, only changing a single variable can be very difficult. Right? So there oftentimes can be confusion in the results based on unknown independent variables that we weren't considering when we actually set up the experiment. Controlled variables are other things that could affect the results that you want to kind of keep the same, so they don't affect the results, right? So if we're looking at our tomato plant example, controlled variables, like we're studying fertilizer levels, our controlled variables might be the amount of sun they get, because that could affect plant growth. Controlled variables could be the amount of water they get, because that could affect plant growth. It could be the type of tomato plants you use, because that could affect plant growth. Right? So there are all the kinds of other things which could affect the dependent variable that we don't want to affect the dependent variable. We're just interested in fertilizer. That's the only thing we want to alter in our experiment. All right, so that's where the other possible variables hopefully are ruled out in this process. And also where we look at controls. If we're doing our tomato plant example, a good control group might be one that gets exactly the same conditions as our fertilizer plants, only instead of fertilizer, they just get water. You treat them exactly the same otherwise. The only thing you don't give them is fertilizer. And why do you want to do that? Why do you want to have a control group? Well, this is kind of like your baseline group. This is your comparison group. This is the one that you're going to compare your treatment groups to see how much difference there was. Okay, so if your non-fertilizer plants grew 5 centimeters and your fertilizer plants grew 8 centimeters, then the difference between the two, difference between 8 and 5, should be the fertilizer. Right? That was the only thing that was different between the two groups. Otherwise, they grow exactly the same. So the control group is your baseline comparison group. Okay? All right. Limitations here of the scientific method. Number one, we already kind of addressed this, is that you can never really truly make sure that all untested variables are controlled. Maybe in growing tomato plants in a greenhouse, maybe there was an insect outbreak that only affected some of the plants. And just because you got bad luck, they only affected the ones that were not given fertilizer. So maybe fertilizer had absolutely no effect, but the fact that the ones that didn't get fertilizer also got fed upon by insects made them grow less. So you might conclude that fertilizer actually helped, even though it didn't, even though it was the insects that actually hurt the non-fertilizer plant. So those are the kind of wrinkles that kind of get thrown in sometimes that can kind of throw you off. There is no proof in science. Now that is something that's oftentimes hard for people to accept or understand. There is no proof. Just because the results come out one way doesn't mean they will always come out that way now and forevermore. Conditions change. Okay, so even though you show the results to be one way, you say that, okay, well, for now, this is what they are. Other conditions, other times, they might come out differently. The next thing that we have to understand with the scientific method, and publishing especially, is that if you go to publish an experiment or results of an experiment, you have to communicate openly and honestly with the reader how you did your experiment. Okay? There is something called the methods section or a materials and methods section where you have to tell the reader exactly, in painful detail, how you did your experiment, right down to the company you ordered your supplies from. So that if they want to, they could actually go back and repeat your experiment to see if the results were valid. If you just say, hey, I got these really cool results, but I'm not going to tell you how I did it, then nobody's going to believe you. You have to communicate openly about how you did what you did. It's part of that quality control process. All right. The other part of things here is that sometimes human personality traits play a role in the scientific method. The best experiments are not only simple, they're also often very creative. And sometimes even luck plays a role. The discovery of penicillin was a dumb luck accidental discovery that somebody made as a result of bad microbiological technique. They discovered antibiotics. It was luck. And kind of an open mind that led them to that discovery. So human personality traits also play a role. Okay, so we've already talked about scientific theories. Theory with a big T, almost like a principle or law. Theory with a little t is like an everyday theory. Right? That's not something that's been proven or even tested yet. So we want to make sure we understand the difference between scientific theories versus everyday theories. Scientific theories have already been tested a lot. They've held up pretty well. They're as close to definitive as a principle or law can be, at least in biology, but it doesn't mean they're set in stone. They can change. If evidence comes along to the contrary, to suggest that a theory is not quite accurate, the theory can be reworked and modified so that it's more accurate. It's not set in stone. It's modifiable if enough evidence comes along. So an example of this, prions. Prions are a discovery that goes back to late 80s, early 90s in biology. This is a type of infectious protein. It's not a living organism, it's a protein. It's a macromolecule, I guess you could say, that is infectious. So prior to the discovery of prions, people thought, doctors thought, the scientific world thought, that only living things could cause communicable disease. Meaning that if you're sick, you can pass whatever is making you sick on to the next person and make them sick. But that only works if the pathogen is a living thing. Prions are non-living. But prions are transmissible, communicable. They can be passed from one host to the next, even though they're non-living. So prior to the discovery of these molecules, people had this idea, this theory, this big T theory, that infectious disease was only caused by living things. That, to that point in time, was as close to a principle or law as you could get. But after the discovery of prions, that had to be reworked. Because it turns out there are non-living things which can cause transmissible infectious disease, like prions. All right, the last section here deals with an example of natural selection. So we have these orchids with very, very long, what are called nectar tubes. And we have these hawk moths, which we actually have relatives of these things even in this part of the world. These are tropical species. But in this part of the world, we have similar hawk moths. They look kind of like a hummingbird, big, meaty kind of moths, that have these really, really long mouth parts that can stretch down into these nectar tubes and feed on the juicy goodness inside. And in the process, they're going to help pollinate these flowers. It's kind of a win-win. The moths get food, the flowers get pollinated, and everybody's happy. Okay. All right, so this goes back to the idea of evolution and what Darwin called descent with modification. Darwin didn't actually use the term evolution. He didn't like it for various reasons, but he talked about descent with modification. So it turns out he didn't invent this idea, something that sometimes surprises people. Darwin was one of many people, like those before him, that talked about this idea, about species changing. Aristotle talked about species changing over time. Darwin's grandfather wrote a book about species changing over time. There was a guy by the name of Lamarck that talked about species changing over time. So evolution was not a new idea. What Darwin did was he kind of came up with a mechanism. He came up with a how of evolution, which prior to his time, people really didn't have a good idea about how evolution worked. So his mechanism, of course, was natural selection, and it's the explanation for why evolution works the way that it does. It's natural selection. So on his voyage around the world on the Beagle, and at this point in time, Darwin was a kid, and he was like 22 years old. He was along for the ride because his dad was rich and he didn't know what he wanted to do with his life. So he got to go on this boat ride around the world, and in the process made a lot of observations. He wasn't trying to prove evolution. He wasn't even thinking about evolution. He was 22 years old. He was a guy. He didn't think about that kind of stuff. He thought about drinking and girls and shooting at stuff and having a good time. That's pretty much where he was at his point in life, in that point in his life. All right, but he did notice things on this voyage. The Galapagos finches, for example, on the Galapagos Islands, very, very similar birds in some way. Not a big stretch to say they might be related to each other, but in other ways they're very, very different. And later on in his life, when Darwin started to think more about science and those kinds of things, this is one of the big lines of evidence he used to talk about how natural selection might work the way that it does. One of the other big things that Darwin worked with on, actually after the voyage of the Beagle, was an essay called On the Principle of Population by a guy by the name of Malthus. Malthus had this idea that there are too many people on the planet. And he wrote this back in like 1796. And at that point in time, there were less than 1 billion people on the planet. So there really weren't that many people compared to what there are now. We're like 7.2 or 3 billion people now. But Malthus said there's too many people because people's populations tend to grow geometrically or exponentially. The blue line represents how people grow, Malthus said. On the other hand, Malthus said, humans are only capable of increasing their food supply linearly or arithmetically, the red line here. So the question for Malthus is, okay, what happens when you're out here with this much food and that many people? And Malthus said, oh, this is not going to be good. We're going to have wars and famines and pestilence and starvation, all kinds of really bad stuff because we have more people than we have food. And he's probably right at some point in time. But what this did for Darwin was this gave him kind of a basis for thinking about natural selection. He said, well, what happens if this occurs in nature? What happens if there are more individuals born than can be supported? If there's more animals than there is food, then clearly some of the animals aren't going to make it. Can we predict which ones might be able to survive? Yeah, it's going to be the ones that have the best traits. The ones that have the best ability to find food and survive and reproduce and pass on their genes. Those are the ones that are going to make up the next generation, their genes. And so eventually that's what became known as natural selection. Evolution is a theory with a capital T. It's not just a theory, as you'll sometimes hear from critics about evolution, people that don't really fully understand it or don't want to understand it. It is a scientific theory. It has been tested literally tens of thousands of times over the past 150, 160 years. And it's held up pretty well. Evolution is based on variability in a population. Individuals have to be somewhat different from each other. If everybody looks exactly like everybody else or behaves exactly like everybody else, then evolution doesn't work. There has to be variability. There has to be some sort of heritable nature of this variability. If it's variability that the environment causes, it doesn't count. If you've got some just outstanding, amazing tattoo that somehow increases your survival and reproductive success, that doesn't count because it's not heritable. When little Timmy is born, when your offspring is born, little Timmy is not going to be born with that same tattoo. So that trait doesn't count for natural selection. It has to be heritable variation. It has to be able to be passed on from parent to offspring variation. And then, of course, there also has to be natural selection. There has to be some type of environmental force that makes some individuals survive and reproduce better than others. So they pass on their genes more than others. So we're going to talk about that variability for a second. The ultimate source of all genetic variability is mutation. Mutations are random. First and foremost, they're random. Changes in information content in the DNA. How do mutations happen? They can happen because of external forces like gamma rays, like what happened to this. This is the old school Hulk, by the way. This is Lou Ferrigno Hulk. This is not modern day Avengers Hulk. This is the old school Hulk. All kinds of chemical, radiation kinds of things can cause damage to DNA, which leads to mutations. Okay, that's one possibility. There can also be, maybe less exciting, are copying mistakes. That when we copy our DNA before our cells divide, sometimes those cells make mistakes. It's not very often. It's 1 out of every 10 million attempts or so. But out of every 10 million tries, you make about 1 mistake copying DNA, so you get that one rogue piece of DNA which doesn't really look quite right. That's a mutation as well. Let's think about what happens with these mutations. The single most common outcome of any mutation is absolutely nothing. It's harmless. It's neutral. It causes no effect on the individual. So maybe 75, 80% of the time, mutations have absolutely no effect whatsoever. The DNA changes, but there is no net effect on the survivability or reproducibility of that individual. That's kind of disappointing. Most mutations are neutral. Not very exciting. Second most common outcome of mutation is some sort of decrease in functionality. It doesn't kill them necessarily, but it makes them less able to grow or survive or reproduce than those individuals who don't have the mutation. So that's a decline in their fitness, in a sense. So maybe, 10% of mutations might be harmful in some way. Okay, so now we're at, 90% mutations? Most of the rest of the mutations, maybe the next 7 to 9% of the mutations, are going to be death. Fatal mutations. Mutations that are experienced in really, really important bits of DNA, that if you change them at all, they fail to work. That would result in the death of the organism in some way. Maybe immediately, maybe later on in life, but it's a lethal type mutation. And that's going to leave us like less than 1% for the really cool mutations. The mutations that give you superpowers, right? Mutations that give you laser shooting out of your eyes and adamantium claws, although that wasn't really a mutation, I guess. But, those kinds of things. Those mutations are really uncommon. I know this because I've never actually seen anybody that could shoot lasers out of their eyes. It's got to be rare, right? But in all seriousness, beneficial mutations are not all that common. They're extremely rare, and even when they do occur, they are tiny, tiny, tiny little mutations. They cause very, very small changes in survivability and reproducibility. Okay, so really uncommon. So all this together means that these changes that occur, this evolution takes a really long time in most cases. It's not an overnight kind of a process. It takes long, long periods of time. Even for things like bacteria, which reproduce really quickly, it can take a long time. All right, so we talk about adaptations. Natural selection tends to favor the genes that result in adaptations. Things that are physical, morphological things that are physiological, process-based things inside your body. Maybe you have a mutation that results in a slightly more efficient enzyme. Maybe it's a behavioral mutation. Maybe it's a mutation that allows you more camouflage, those kinds of things, right? So there are lots of different kinds of mutations that can lead to adaptations, but the thing to remember here is that the adaptations are contextually dependent on the environment. I'll give you an example of that, right? If you've ever thought about why people have different colored skin, maybe you have, maybe you haven't, but there is a really good explanation for this. When Homo sapiens first evolved, it was in Africa, equatorial region. Think about the places in the world where typically people have really dark colored skin naturally. They're all equatorial places. South America, Central America, Africa, Southeast Asia, South Asia for that matter, and Northern Australia. Dark colored skin. And there's a good reason for that, because if you have dark colored skin in those environments, you have more protection against UV radiation, which can be bad for you. So people evolve in Africa. They evolve with dark colored skin. They move out of Africa. They go all around the world, including places like Northern Europe. Now, if you know anything about people from Northern Europe, you know they don't have dark colored skin. So apparently at some point in time, there was a selective process that led people in Northern Europe to stop having dark colored skin and start having light colored skin. Mutations popped up in the human population that led some people to have lighter colored skin. And for whatever reason, in Northern Europe, lighter colored skin was favored. Darker colored skin was favored in Africa. Light colored skin was favored in Europe. Now, why is that? Well, it turns out that if you have dark colored skin, not only does that help block out UV, which is great. I mean, I totally wish I had darker skin in the summertime or when I've been to the tropics. I need dark colored skin because I can put on SPF 1000 and I also get sunburned, right? So in the tropics, dark colored skin is your best friend in the world. But if you live in a really far north or really far south kind of place away from the equator, you run into problems with vitamin D deficiency if you have dark colored skin. Because one of the things you need to have for your body to make vitamin D is you need to have sunlight absorption by the skin. If you've got really dark colored skin with a lot of melanin in your skin, you're going to have a hard time absorbing that light to make that vitamin D. And so you run into vitamin D deficiency. If you have light colored skin in Finland or Norway or Sweden, then you're able to absorb lots of vitamin D. Okay? So in those parts of the world, there isn't that much solar radiation relative to the tropics. You're better off to not filter out sunlight from your skin. So that the point of this story is that in one part of the world, dark colored skin is favored, the tropics, In a different part of the world, dark-colored skin is selected against because of the amount of sunlight they get and the lack of vitamin D that's produced because of the sunlight or lack thereof in those places. All right, so we need to talk about this term fitness. We tend to think of, whenever I ask the question about natural selection, people always want to say survival of the fittest. But what I find is people don't really understand what fitness is. It's not fitness in like a go to the gym kind of fitness sense. It's fitness in an evolutionary sense. Fitness is your survival to grow, survive, and reproduce. And sometimes, yes, that does mean being bigger and stronger and having bigger fangs and pointier antlers and whatever. Sometimes fitness does mean big and strong. But sometimes fitness doesn't. Sometimes fitness means better camouflage. Sometimes fitness means more efficient enzymes. They're able to carry out processes using less energy in your body. That could be a fit kind of thing to have. So you got to be careful about what fitness means. Maybe fitness means plants that are able to transport water more efficiently. Not very exciting. It's not like, big, strong, muscular lions chasing giraffes or zebras or gazelles or those kinds of things. It means plants transport water better. But that might make that plant more fit than another plant. Okay, so fitness is a relative kind of a thing. A really good example of natural selection is a classic example going back to the 1950s. The left-hand picture here, there are two moths. There's a moth here on the left, dark-colored. Here's the head, here's the abdomen, here's the left wing, here's right wing. There's another moth right here. I'll sort of outline it like this. And there's a moth there. You kind of see it now. Here's the head, here's the abdomen, here's the left wing, kind of like a triangle, and here's the right wing, kind of like a triangle, right? Really well camouflaged. So when this picture on the left was taken, there were a lot of these white kind of crusty things growing on the sides of these trees called lichens. Not the werewolf kind, the other kind. These are lichens. And these light-colored moths are really well camouflaged on those lichens. Dark-colored moths, not so much. Those kind of stick out like a sore thumb. So if you're a bird flying around in this forest, you're going to find a really, really easy time tracking these down and a much harder time tracking down the light-colored moths. This, by the way, is the same species. These are just different forms of the same species. Dark-colored form, light-colored form. In the natural environment, the light-colored form makes up like 95% of the population. But what occurred in the study system was that air pollution in these forests killed off these lichens, darkened the trunks of the trees, made them kind of sooty and dark because of the ash and stuff that was in the air. And so all of a sudden, the tables are turned. Now the dark-colored moth is camouflaged, the light-colored moth sticks out like a sore thumb. And over the span of like a decade, the percentages were switched. About 10 years after this change was noted, the dark-colored form made up like 90% of the population, and the light-colored form made up like 10% of the population. And then after Great Britain implemented air pollution controls, we see a switch that occurred. Right? So we see a switch from 90% light-colored moths back down to about 10% light-colored moths over the span of, 35 or so years. So conditions in the environment determine which individuals grow and survive and reproduce the best. In other words, it affects the fitness of the different forms. All right, so that's the end of our chapter 1. I'm going to cut this off here. As always, make sure you email me if you have any questions or concerns, and I will do my absolute best to try to help with those.